Okay, in this next video, we're going to look more carefully at muscular contraction. And so to kind of get our head back in the game, I just have this first picture to remind us of what the muscle looks like. So when you look at a whole muscle, the whole muscle is going to be wrapped in this layer called the epimysium. And that epimysium is really connecting and surrounding these fasciculi, so these muscular fasciculi. Each muscular fasciculi is going to be surrounded by this layer of connective tissue called perimysium. And inside each of these fasciculi will be the individual muscle cells, which are also called muscle fibers. And so if we pull out an individual muscle fiber, for one, it's going to be surrounded by endomysium. But then inside this muscle fiber are going to be many of these packages, which are called myofibrils. And what each myofibril is it's going to be full of these long, skinny proteins called myofilaments. And the myofilaments are going to be actin and myosin. And in the last video, we kind of looked at how actin and myosin are arranged into this repeating subunit called a sarcomere, with the Z discs connecting the actin, and then myosin being connected to an M line and kind of being scattered in between. So we're going to pick up here and look more carefully at how the sarcomere actually goes about contracting. So here is another image of first the myofibrils with all of its myofilaments inside, and these myofilaments include both actin and myosin, and they're arranged into these repeating subunits called sarcomeres. So here, this is an example of a sarcomere. And so in this image, I want you to see the Z disc, and this Z disc is going to have the actin attached to it. So these little purple bits are representing the actin. And then it's kind of showing you the M line right here, and then attached to the M line is going to be the myosin. Okay, so what we can add now to what we know about these is going to be that this myosin actually has these little bits called myosin ores. And so that's what these are right here. They're called myosin ores, or they could be called myosin heads. And so in this picture here, we're able to see the myosin myofilament a little bit better. Here's the myosin, and at the end of it, it's got these little extensions, which are called the myosin heads. The idea is that these myosin heads will be able to attach to the actin. And these heads are going to be able to bend and straighten using ATP. So these heads are going to be really important to the ability of the muscle to contract. We're actually going to watch these heads grab hold of the actin and then bend. And as they bend, they're going to push the actin towards the middle. They're going to be pulling this actin, which is pulling on the Z discs, and this will actually pull those Z discs toward the M line, contracting the muscles. So these myosin heads are going to be able to grab the actin and pull them toward the middle or toward this M line. Okay, so that's the myosin. Now, the actin is the other one that we're seeing here. So it's the one in purple. The actin, of course, has binding sites for these myosin heads to bind to. However, these binding sites are not always available. So here in this little picture down here, we're kind of zoomed in on the actin with these purple things. You know, we can see the purple blobs, which are the actin. And we're able to see that the actin has this molecule called tropomyosin. And what tropomyosin does is it blocks the myosin binding sites. So in this picture of our actin, the tropomyosin is this blue, I'm going to kind of get a blue color here, it's this blue protein that wraps around the actin molecule, or the actin protein. And what it does is it covers up the binding sites for myosin. So this word tropomyosin, in my mind, I kind of like to say it's trips up myosin, because that's what it does. The tropomyosin trips up myosin, or makes it so that the myosin cannot attach. Okay, now the tropomyosin is going to interact with this other molecule called troponin. And so troponin is indicated here as kind of these little red blobs that are also attached to actin. And troponin is interacting with the tropomyosin, but troponin is the player, our little protein molecule player, that has the ability to bind calcium and move tropomyosin. So, here's the full story. When there is no calcium around, the tropomyosin covers up these binding sites and myosin cannot bind to actin. So if there's no calcium, tropomyosin trips up myosin and makes it so that it cannot bind to the actin. And this would make it so that there's no interaction between myosin and actin if myosin cannot bind actin because of tropomyosin being in the way. In which case, these filaments, which are called sliding filaments, because they're going to slide past one another, if myosin cannot bind the actin, then they're just freely able to move, like if you were to apply gravity or apply an opposing muscle, these sliding filaments would just slide along because they're not actually touching or bound to one another. However, in the presence of calcium, so we have to talk about how calcium gets here. A little spoiler is it has to do with the action potential spilling the calcium from the sarcoplasmic reticulum, but we've not gotten there yet. So without calcium, tropomyosin's in the way. And the myosin and actin cannot interact, and so they cannot cause any kind of contraction. But if calcium is released out here to this part of the cell where the myosin and actin are, if we release calcium and now there's calcium around, calcium will bind to the troponin, which will push the tropomyosin out of the way. So that's written here. In the presence of calcium, troponin will bind the calcium and move tropomyosin off the binding sites. Said as simply as possible, in the presence of calcium, myosin binds actin. And when myosin binds actin, these myosin heads may be able to move. We need to talk about how these myosin heads move. Basically, they just need ATP, and then these myosin heads would be able to move. But the thing that I'm trying to focus on right now is calcium. And so in order to have the myosin heads bind the actin at all, calcium has to be present, because calcium binds to troponin, and troponin, when bound to calcium, will move the tropomyosin so that it's no longer blocking the ability of myosin to bind to actin. Whew. And then when myosin can bind to actin, myosin can do its job, which is to use ATP to bend and straighten. Okay, so now that we have a little bit better understanding of the myosin and actin, let's look back at their arrangement in the sarcomere. So the sarcomere is going to run from Z disc to Z disc, and it's going to be many repeating units within a whole muscle fiber. So within these myofibrils that are within the muscle fiber, you're going to have, along the length of one myofibril, you're going to have many of these Z discs and many of these sarcomeric units. So there's going to be this repeating subunit. In this picture, we're kind of seeing two of them. And this could happen in all of the sarcomeres at the same time, because within a whole muscle fiber, if calcium is released and now there's calcium in this space near the sarcomeres, then calcium will interact with the troponin, which will move the tropomyosin and allow the myosin to grab the actin. And then what we're going to see happen is these myosin heads are going to bend, and as they bend, they're going to pull the Z disc toward the M line, which is right here in the middle. It's what the myosin filaments are attached to. And that's going to happen over here as well. These myosin heads are going to bend and pull that actin toward the midline, moving that Z disc toward the midline. And that's going to happen over here as well. This is going to be pulled toward the midline. And that is how the muscle cell is going to shorten along its whole length. All of these sarcomeres will do it simultaneously. I will be linking in some of these YouTube videos because it's really hard to draw. It's something that you need to kind of watch as it happens. But I do my best, and so in these images you can kind of see that contraction happening as these as the myosin pulls the actin this way and pulls the actin this way, what we see is there is more overlap between the myosin and actin. So this becomes something really important to understand. When we talk about muscular contraction, the myosin myofilaments and the actin myofilaments do not change length. The myosin and the actin in all of these pictures, this little purple molecule and this little green molecule, are the exact same length. All that's happening when the muscle contracts is the amount of overlap between the myosin and actin increase. So down here in the bottom image where we have the most contraction, there is complete overlap between this purple actin molecule and the green myosin molecule. They're completely overlapped. There's no area where there's myosin alone or actin alone. And especially if you go back and you compare to where we started with, up here, this was the relaxed muscle in the top image. And in the top image, there's this great big area of just myosin all by itself with no overlap. And there's actually the same thing for an area of actin with no overlap. And so what's happening as we contract the muscle is this amount of overlap is increasing, and that's why the muscle shortens. So I think this is one mistake that a lot of people make, is they forget myosin and actin, the proteins, the myofilaments, myosin and actin, will never change their length. So then how does the muscle cell shorten? The answer is they increase their overlap. And that's what makes the cell shorter. Okay, so what I'm going to try to do next is kind of go through the step-by-step story of what happens in muscular contraction. In the next video, the third video, I'm going to talk more about the action potential. But I think you already know a little bit about it, as far as an action potential is going to be this change in voltage that's going to spread across the cell membrane. So here's that word sarcolemma, which means cell membrane. And remember, the cell membrane invaginates as what are called T-tubules. So when the action potential, which is a voltage change, spreads along the cell membrane, it also spreads into the T-tubule. And then if we kind of look at this down in the picture, here's the cell membrane. So we're going to have an action potential spreading across the cell membrane. But also, as it spreads across the cell membrane, it's going to spread into the T-tubules. So we're going to have this change in voltage going into the T-tubule. So it's spreading across the whole cell membrane, and we're having this voltage change happening within the T-tubules. The T-tubules, if you recall, are right next to the sarcoplasmic reticulum. The sarcoplasmic reticulum is the new name that we have for endoplasmic reticulum, but it's really unique in the muscle cell because it's these hallway systems. So you're kind of seeing the sarcoplasmic reticulum as these flat cisternae, or these flat hallways, and they are full of calcium. I'd kind of given you as at a sneak peek. Now I'm telling you it for real. The sarcoplasmic reticulum is sequestering all of the calcium. So at rest, all the cell's calcium is locked inside these cisterna called the sarcoplasmic reticulum. That means that there's lots of calcium inside these blue hallways, which are the sarcoplasmic reticulum, and not a lot of calcium out here around the myofilaments. So the actin and myosin, at rest, they have no calcium around them because all the calcium is locked inside the sarcoplasmic reticulum cisternae, or those inner compartment. So what's going to happen with this action potential, as the action potential spreads down the T-tubule, it's causing a change in voltage. That's what an action potential is. It's this change of voltage that's happening along the T-tubules. The sarcoplasmic reticulum membrane has voltage gated, so that means that this voltage change is going to open these voltage gated calcium channels. And when you open these voltage gated calcium channels, now the calcium that was trapped is going to be spilled out. And now we're going to have calcium out here with our myofilaments. So the action potential causes the sarcoplasmic reticulum to spill the calcium. And it does that because the sarcoplasmic reticulum has voltage gated, meaning that it's gated by the voltage, and that change in voltage, which is the action potential, will open these calcium channels, allowing the calcium to come out. Now, when calcium comes out, we have now calcium around the actin and myosin. So remember, before it wasn't there. There was no calcium out here. But now that we've spilled it, now we have calcium near our myosin and actin. Also remember that when there's no calcium, tropomyosin covers the binding sites on actin so that the myosin heads cannot bind it. These guys cannot touch each other. The tropomyosin's like, can't touch this, and the myosin can't touch it. But as soon as you spill the calcium, calcium goes off and interacts with troponin, and troponin pushes that tropomyosin out of the way. So troponin pulls on tropomyosin, revealing the actin's binding sites for the myosin heads. And now we have myosin able to bind actin. This is called forming the myosin cross bridges. Now we have connectivity between our myosin myofilaments and our actin myofilaments. Without calcium, that tropomyosin blocks it. But as soon as we spill the calcium interacts with troponin, and troponin interacts with tropomyosin, pushing tropomyosin out of the way, and then the myosin heads can bind the actin. But we needed the action potential to cause the voltage change that would open those voltage-gated calcium channels to get the calcium released. So the action potential causes the spillage of calcium, and then the calcium, it causes the myosin and actin to interact. So this is sometimes called action potential and contraction coupling the voltage change to muscular contraction. Okay, so now that the myosin can bind the actin, how does it actually cause them to slide along one another in order to bring the Z discs closer? And the answer is, well, ATP. So you need ATP to cause these myosin heads to bend. And actually, ATP was already here. We're going to talk about when the ATP got there. But there's some energy stored in the myosin heads already. So as soon as the myosin heads are able to lock onto the actin, they bend. And this is called the power stroke. As the myosin heads bend, they pull those actin toward the M line, or pull the Z discs, which are attached to the actin, toward the end line or toward the middle, narrowing the distance between the two Z discs and causing the muscle cell itself to become shorter. Okay, so then what has to happen though, so each of these myosin heads are really small, and so when they bend, we're moving this thing, micrometers along. So what has to happen is these myosin heads need to release and bind again. And this needs to happen over and over. We need to have the myosin heads let go, recock is what it's called when they kind of straighten back out, so that they can then bend again. So the next step is ATP. This is our first ATP really, because before I said there was an energy stored from an ATP. This is where it came from. A new ATP binds the myosin heads. And this ATP provides the energy for the myosin heads to release actin. So there was stored energy for the myosin heads to bend, but then we need a new ATP. So this is kind of the first ATP we've talked about. We need an ATP to bind and break down. So as the ATP cleaves off one of the phosphates, it becomes an ADP. So the T is for 3, the D is for 2, and we're talking about numbers of phosphates. So when you break down ATP, you form ADTP and a free phosphate. So this is energy is released here as well. So this is breaking down ATP to release the energy. ATP is broken down in order for the myosin heads to let go of the actin. And then once the myosin heads let go of the actin, you can see they've got a little bit of energy store here that they're going to use. But the idea is, and then they go back to resting position, and they've got that energy stored. The idea is if the calcium is gone, then they won't rebind. But generally, there's still calcium around. We're going to talk about getting that calcium cleaned back up. But if that calcium is still around, then the tropomyosin is still out of the way. Then the myosin head will bind again, and as long as there's still ATP, it will bend and release again. And then as long as the calcium's still there, getting the tropomyosin away, and there's still ATP there for it to let go and recock, it will bind again, bend again, and as long as there's still ATP, it'll release again. And then as long as there's still calcium, it'll bind again. And it'll bend and pull. And then as long as there's ATP, it'll release. And we do that over and over. And so this part, you really want to watch a video to see these guys bending and releasing. And so I'm going to link in several videos so that you can see that happening live. But this little slide kind of sums up main idea here. As long as calcium is out there and attached to troponin, those binding sites are available. So myosin will bind to the actin. All that's required for myosin to bind to actin is the availability of calcium. And then all that's required for the myosin heads to bend and pull the actin toward the Z disc is the availability of ATP. So ATP is needed to move the heads and thus move the actin, because if we move the heads, we move the actin that it's attached to, and we need calcium so the myosin can bind the actin. We need ATP so the heads can move. So those are the two ingredients that are necessary during muscular contraction. And then finally, calcium, once it gets spilled, that calcium stays out there in the sarcoplasm until it is actively pumped back. So this requires ATP. If you don't have ATP, we can't re-sequester the calcium back into the sarcoplasmic reticulum. So calcium stays out in the sarcoplasm interacting with myosin and actin until enough time and ATP has passed in order to get all the calcium re-trapped in the sarcoplasmic reticulum. So that requires, the voltage change is gone. There's no longer an action potential happening on the muscle cell to open the calcium channels. And then there's going to be an ATP-powered calcium pump that actually picks up the calcium and pumps it back into the sarcoplasmic reticulum membrane. This requires ATP. And then once you get that calcium cleaned up, and back into the sarcoplasmic reticulum, then the tropomyosin will move back over and cover those binding sites on the actin, and then there'll be no more interaction between the myosin and the actin. Okay, so it seems like a lot, so I'm just going to say the same thing, and by repeating myself, I don't want you to feel like I'm saying new information. The same information, I'm going to say it one more time. Calcium is sequestered in the sarcoplasmic reticulum membrane until a voltage change travels along the membrane and up the T-tubules, and we get this voltage change right next to the sarcoplasmic reticulum membrane. On the sarcoplasmic reticulum membrane are these voltage-gated calcium channels. So when this voltage change happens, these calcium channels open and calcium comes out. When calcium comes out, the calcium is able to bind to the troponin that interacts with the tropomyosin, and these tropomyosin molecules move out of the way so that we now have these free binding sites for the myosin to bind to the actin. So myosin's able to bind actin only in the presence of calcium. Then, if ATP is available, the myosin heads bend and pull the actin toward the Z line, release, and as long as calcium's still there, they bind again. And then they repeat. They bend, release, and as long as calcium's still there, they bind again. So in order for this to happen, we need two things. We need calcium to be near the myosin and actin, and then we need ATP. If there's no ATP, what would happen is the myosin grabs hold of the actin and cannot let go again. And we need it to let go so that it can recock and bind again, or bend again. So we need ATP to move the head so that they can release, cock, recock, kind of go back into that unbended formation. Release, cock, bind, and bend again. And then this has to repeat. And you need an ATP for every cyclic repeat of that. And then in order to stop the muscular contraction, we have to use ATP to pump the calcium back away. And then as soon as we get the calcium all pumped back up, the tropomyosin recovers the binding sites, and myosin can no longer interact with actin. The cell is back at rest again. So that gives you an idea of how muscular contraction happens. And as I said, there's going to be several videos linked in as well that kind of show you this happening in real time.